Welcome to the deep dive. Today, we're really going beyond a simple summary. We're aiming for, well, a masterclass, tackling a pretty complex stack of source material all about the male reproductive system.
Yeah, this is about getting that granular, professional-level understanding. Histology, physiology, the works.
Exactly. We want you to feel like you've truly mastered it by the end.
So our plan is to break it down systematically. We'll cover the testes, obviously, then the genital ducts, the accessory glands, and finally the penis. All looking at the cellular mechanics, the hormones, the clinical side, too.
That's the mission. Not just overview, but precision. The kind of detail that really separates foundational knowledge from, well, elite professional understanding. We're linking structure to function at every step.
Yeah.
Maybe the best place to start is right with the tests themselves. They have this dual identity, really.
Meaning?
Well, everyone knows they produce sperm, the exocrine function, but they're also incredibly potent endocrine glands
Yeah, I heard.
producing hormones.
Primarily testosterone. It's the main driver, for overall male physiology, for spermatogenesis itself, and absolutely critical for sexual differentiation way back in the embryo.
And the sources also really highlight testosterone's metabolite, dihydrotestosterone, DHT. It's not just testosterone doing everything, is it?
No, definitely not. Think of testosterone as like the main engine. DHT is more specialized, particularly post-puberty.
How so?
It acts locally, a really potent signal, regulating growth and function in specific places. Think accessory glands like the prostate, and also secondary sexual characteristics, hair follicles, that kind of thing.
So T for the core functions, DHT for specific pubertal tuning.
That's a good way to put it. Understanding that split is pretty fundamental.
Okay, let's ground ourselves in the anatomy first. The tests themselves. Structurally, they seem built for protection, encased in that thick layer, the tunica albuginea.
Right. It's a dense, white, sort of fibroelastic capsule, very tough.
And it's not just a simple covering, is it? It actively shapes the inside.
Exactly. It thickens up significantly on the posterior side, forming what's called the mediastinum testis.
Okay, the mediastinum.
Yeah, think of it as the central hub. It's the entry and exit point for blood vessels, lymphatics, and importantly, the ducts leaving the testis.
And from that mediastinum, walls project inwards, septa.
Correct. Connective tissue septa divide the whole organ into these compartments, roughly 250 of them, pyramidal-shaped testiculi lobules in each testis.
Wow, 250. That's quite a few.
It is. And inside each lobule, that's where the action happens. You've got, say, 1 to 4 highly coiled seminiferous tubules. That's where sperm are actually made.
And between those two.
Sparse interstitial tissue. It looks sparse, but that's where you find the crucial Leydig cells that make the testosterone, plus blood vessels, lymphatics, etc.
Now, the location. This is key. They start inside the body, retroperitoneally, but have to descend into the scrotum.
Yes, and that descent is fascinating. They essentially drag a piece of the abdominal lining, the peritoneum, down with them.
Which becomes
The tunica vaginalis. It's a serous sac that partly surrounds the testes within the scrotum. Clinically, that's the space where fluid can build up a hydrocele.
okay, that makes sense. But the reason for the descent, for being outside the main body cavity, is all about temperature, right? This 34 degrees C rule.
Absolutely non-negotiable. The core body temperature, around 37 degrees C, is simply too high.
Too high for what, specifically?
For the molecular events of sperm formation, particularly the later stages, meiosis, and especially spermatogenesis, that final differentiation step, they just don't work efficiently, or sometimes at all, at 37 degrees.
So the scrotum's job is to maintain this cooler environment, around 34 degrees C. How does it pull that off? There are a few mechanisms.
3 main ones, yeah. The first is just ingenious vascular plumbing, the pampiniform venous plexus.
Sounds complex.
It is, visually. It's this incredibly dense network of veins wrapped tightly around the incoming testicular artery.
Okay, veins around the artery. Why?
It creates a countercurrent heat exchange system. Think about it. Warm arterial blood at 37 degrees C flows down towards the testes, while cooler venous blood at maybe 34 degrees C is flowing away, right next to it.
So the heat transfers.
Exactly. Heat flows from the warm artery into the cooler surrounding veins before the arterial blood even reaches the sensitive testicular tissue. It's pre-cooled on the way in.
Wow, okay, that's clever. An anatomical radiator.
Pretty much. Mechanism 2 is simpler, just evaporative cooling. Sweat evaporating from the scrotal skin helps dissipate heat.
Right. And the third is active muscular control.
Yes, dynamic adjustments. You've got two muscle sets involved. First, the ductus muscle, which is a thin layer of smooth muscle within the scrotal wall itself.
And the cremaster muscles.
Those are skeletal muscle strands running within the spermatic cords, attaching to the testes.
What do they do differently?
When it's cold, they work together. The ductus contracts, wrinkling the scrotal skin, reducing surface area. The cremasters contract forcefully, pulling the testes up closer to the warmth of the body.
And when it's warm?
They both relax. The scrotum becomes smooth and hangs lower, maximizing surface area for cooling and moving the testes further from the core body heat. It's a constant fine-tuning.
Truly dynamic regulation. Okay, let's go back inside to that interstitial tissue between the tubules.
Yeah.
Where the testosterone gets made.
Right. The space contains fibroblasts for structure, lymphatics, but critically, it has fenestrated capillaries.
Fenestrated meaning?
They have small pores or windows. This allows for really rapid exchange, particularly getting those steroid hormones into the bloodstream quickly.
And the cells doing the making are the Leydig cells, or interstitial cells. What do they look like? The sources mention a typical steroid producer morphology.
They stand out. They're large, usually round or polygonal, with a big central nucleus. The cytoplasm stains pink, eosinophilic, and it's packed with tiny lipid droplets.
Why is that significant? Lipid droplets and pink cytoplasm.
The lipid droplets are cholesterol stores, the precursor for steroid synthesis. The eosinophilia comes from the massive amounts of smooth endoplasmic reticulum and mitochondria needed for all the enzymatic steps to convert cholesterol into testosterone. It's the classic look of a steroid factory cell.
And the production pathway is similar to the adrenal cortex then. What triggers it? It's not just always on, is it?
No, it's under pituitary control, specifically luteinizing hormone LH from the pituitary gland.
Which used to be called ICSH.
Exactly, interstitial cell stimulating hormone, because it targets these Leydig cells. LH secretion ramps up at puberty, binds to receptors on the Leydig cells, and flips the switch for testosterone production.
But wait, the sources mention fetal Leydig cell activity. If puberty is the main trigger, why are they active before birth?
This is absolutely crucial for development. There's a window around the 3rd and 4th months of gestation where fetal Leydig cells are highly active.
Stimulated?
Placental gonadotropin, HCG, actually.
Yeah.
The testosterone they produce then is essential for masculinizing the embryo, developing the male duct system and accessory glands. Without that early testosterone surge, male development gets derailed, regardless of puberty later on.
So they do their job, then go dormant.
Pretty much. They regress and look quiet until LH kicks in years later at puberty.
Okay, clinically wrapping up this section. Hydrocele, we mentioned fluid in the tunica vaginalis. But cryptorchidism seems much more serious.
It is. Failure of one or both tests to descend happens in maybe 4% of newborns.
And if it doesn't correct itself?
If it's bilateral and not surgically corrected, it causes infertility. The undescended testes is stuck at 37 degrees Curie.
Which damages the sperm-producing cells.
Permanently if left too long. That's why surgery needs to happen early, by age 2 or 3 ideally, to preserve fertility potential.
And there's a cancer link, too.
A significant one. About 95% of testicular cancers arise from germ cells. And the risk of developing these tumors is much, much higher in tests that remained undescended, likely due to the damage from that abnormal temperature and environment. So anatomy, temperature, fertility, and cancer risk, they're all linked.
All right, let's move into the factory floor itself, the seminiferous tubules. The scale of production is just immense. What was it, 200 million sperm per day?
Roughly, yeah, in a young adult. It's staggering. And the tubules themselves, if you uncoiled them from one testis, would stretch about 250 meters.
Incredible. So what's the environment like inside these tubules?
They're lined by a very specialized stratified epithelium, the germinal or spermatogenic epithelium. This rests on a basement membrane, and just outside that is a layer of fibrous connective tissue containing myoid cells.
Myoid cells, what's their role? Are they muscle?
They're like smooth muscle cells, yeah. Flat, contractile. Their job isn't powerful contraction, but rather generating these weak rhythmic pulses.
To do what?
Just to gently move the fluid and the developing sperm, which aren't motile yet, along the winding path of the tubule towards the exit. A little nudge.
Okay. The whole process from start to finish takes about 10 weeks. It starts with spermatogenesis, the division phase.
Right. Begins right at the base, near the basement membrane, with the spermatogonia. These are the stem cells and progenitor cells.
Two types.
Yeah, you have the true stem cells, often called dark type A, dividing infrequently just to maintain the pool. Then you have the pale type A, the transit amplifying cells, which divide much more rapidly by mitosis.
These type A cells then become...
They undergo some unique mitotic divisions to become type B spermatogonia. And it's the type B cells that divide mitotically one last time to produce the primary spermatocytes.
And these are the cells that enter meiosis.
Correct. The primary spermatocyte is a key player. It's the largest cell in the lineage, big nucleus, lots of activity. It's diploid, 46 chromosomes, but it's already replicated its DNA, so it has 4N DNA content.
4 sets of DNA. It depends a long time the first part of meiosis array, right? Prophase.
A remarkably long time, about 3 weeks. This extended prophase is where all the crucial genetic shuffling happens, synapsis, where homologous chromosomes pair up and crossing over recombination. It generates genetic diversity.
Meiosis finishes producing
2 secondary spermatocytes. Now, these are haploid for chromosome number 23 chromosomes each, but each chromosome still has 2 chromatids, so they have 2N DNA content.
And the sources say these are really short-lived, hard to spot.
Extremely transient. They immediately dive into meiosis the second.
Which is more like mitosis.
Kind of, yeah. It's an equational division, just separating the sister chromatids. Because it's so quick, you very rarely catch a secondary spermatocyte in a histological section. It divides almost instantly.
To produce what?
4 spermatids. These are finally truly haploid 23 chromosomes, 1N DNA content, round, simple-looking cells at this stage.
Okay, but here's where things get really interesting structurally.
Yeah.
This clonal nature, the intercellular bridges. Why don't the cells fully separate after dividing?
This is one of the most elegant biological solutions. After that initial spermatogonia stem cell division, all subsequent divisions, type A to B, B to primary spermatocytes, meiosis I, meiosis II, involve incomplete cytokinesis. The cytoplasm doesn't fully divide.
So they stay connected, like beads on a string.
Exactly. They form a syncytium linked by these cytoplasmic bridges. You can have clones of up to maybe 100 cells all connected.
Why? What's the advantage?
It solves the genetic problem of being haploid. Remember, after meiosis, half the spermatids will carry an X chromosome, half will carry a Y.
Right.
But the genes needed for proper maturation might be on the X chromosome, which the Y-carrying cells lack, or vice versa, by keeping everyone connected in the syncytium.
They can share.
Precisely. Proteins, RNA, whatever is needed, synthesized from the complete diploid genome information from both X and Y present before meiosis, can be shared amongst all the linked haploid cells through these bridges. It ensures every single spermatid gets the full toolkit required to mature properly, regardless of which sex chromosome it ended up with.
That is incredibly clever, a biological safety net. Okay, so spermatogenesis gives us these round haploid spermatids all linked together. Now they need to transform into actual spermatozoa. That's spermiogenesis.
Correct. No more cell division now. This is pure differentiation, and it's the really temperature-sensitive part. It happens in 4 phases.
Phase 1, Golgi phase.
The Golgi apparatus gets busy, producing small vesicles, proacrosomal vesicles, which then fuse together to form one large membrane-bound structure called the acrosomal cap, sitting over one pole of the nucleus. Meanwhile, the centrioles migrate to the opposite pole to start forming the axoneme, the core of the flagellum.
Phase 2, cap phase. The acrosome is key here, functionally.
Hugely important.
Yeah.
The acrosomal cap spreads out to cover about half of the nucleus, which is starting to condense. Functionally, think of the acrosome as a specialized lysosome, a bag of powerful hydrolytic enzymes.
Like
2 critical ones are hyaluronidase, which helps the sperm chew through the cumulus cells surrounding the egg, and acrosin, a protease.
What does acrosin digest?
The zona pellucida, that tough glycoprotein coat around the egg itself. Without the controlled release of these enzymes, the acrosomal reaction fertilization is impossible. The sperm simply can't penetrate.
Okay, phase 3, acrosome phase. This is where the nucleus really changes shape.
Dramatically. It elongates and becomes incredibly condensed. This involves a fascinating molecular switch. The usual DNA packaging proteins, histones, are kicked out and replaced by small, very basic proteins called protamines.
Why protamines?
They allow for an even tighter, more extreme compaction of the DNA than histones do. This makes the nucleus smaller, more streamlined for swimming, and protects the DNA during its journey. At the same time, mitochondria gather around the proximal part of the flagellum.
Forming the middle piece?
Right. That's the powerhouse, packed with mitochondria to generate the ATP needed for flagellar movement.
And finally, phase 4, maturation phase.
The final touches. The spermatid sheds all the cytoplasm it no longer needs as a blob called the residual body. This gets immediately gobbled up by the surrounding Sertoli cells. The intercellular bridges finally break down, and the fully formed spermatozoa are released into the tubule lumen.
But not yet modal, right?
Structurally complete, but functionally immature and non-modal at this point.
Clinically, this complexity seems ripe for error. Is this why poor semen quality is a major cause of infertility? Things like oligospermia or morphology problems.
Absolutely. The whole 10-week process is incredibly intricate. Oligospermia, low sperm count, usually defined as less than 10-20 million per milliliter, might reflect problems earlier on, maybe in the mitotic divisions.
But abnormal shapes or poor swimming?
That points squarely at problems during spermatogenesis. Maybe the acrosome didn't form right, or the mitochondria didn't cluster properly in the middle piece, or the flagellum itself is defective. Any hitch in that complex differentiation process can lead to sperm that look wrong or just can't swim effectively, leading to infertility.
Okay, so if the germ cells are the product on the assembly line, the Sertoli cells are. Well, they seem to be everything else. The managers, the support crew, the security guards.
That's a pretty good analogy. They are absolutely essential pillars of the whole system. Structurally, they're quite distinct.
How so?
They're very tall, columnar epithelial cells. They sit on the basal lamina of the tubule, and their tops extend all the way to the lumen. Each Sertoli cell physically envelops and supports a cohort of developing germ cells, maybe 30 to 50 of them.
And how do we spot them on a slide?
The nucleus is the giveaway. It's large, usually irregular in shape, ovoid, or even triangular, and it's euchromatic, meaning the chromatin is open, actively transcribed. Plus, they typically have a very prominent, often triangular nucleolus, very characteristic.
Let's talk about their most famous role, the blood-testis barrier. The sources call it the tightest barrier in the body. How is it formed?
It's formed by incredibly extensive and complex, tight occluding junctions. But here's the key. These junctions are only formed between adjacent Sertoli cells, low down near the basal lamina, not between Sertoli cells and germ cells.
Okay, Sertoli to Sertoli junctions.
Yeah.
Why is this barrier so critical? Why seal off the developing sperm?
Timing and immunology. Think about it. Meiosis produces genetically unique cells, the spermatocytes and spermatids. They have new antigens on their surface that weren't present when the immune system was learning self versus non-self early in life.
Ah, so they'd look foreign.
Exactly. If the immune system lymphocytes, antibodies circulating in the blood, and interstitial fluid could reach these later stage germ cells, it would mount an attack. Autoimmune orchitis, essentially destroying fertility. The barrier creates an immune-privileged site.
So it divides the tubule into two compartments.
Correct. Below the tight junctions, you have the basal compartment. This contains the spermatogonia, which are still deployed and accessible to the interstitial fluid and immune surveillance.
And above the junctions.
That's the luminal compartment. It's isolated, protected. It houses the primary spermatocytes, secondary spermatocytes, and spermatids, all the cells undergoing or resulting from meiosis.
Which brings up a puzzle. The primary spermatocytes start in the basal compartment, but they have to get into the luminal compartment to continue meiosis. How do they cross this tightest barrier without making it leaky?
This is truly impressive cell biology. It's not like they just push through. As a primary spermatocyte linked to its syncytial buddies is ready to move up,
Yes.
the Sertoli cells right next to it have to coordinate. They temporarily disassemble the tight junction strands below the migrating spermatocyte while simultaneously forming new tight junction strands above it.
Wow, so they basically move the barrier itself locally around the cell.
Precisely. They pass the cell through this transient channel without ever compromising the overall integrity of the seal between the basal and luminal compartments. Given that the cells are linked by bridges, it requires remarkable coordination.
Incredible. Okay, beyond the barrier, what are their other main jobs? The sources list 3 general functions. First, support and nutrition.
Right. Since the luminal cells are cut off from blood plasma, they depend entirely on the Sertoli cells. The Sertoli cells have to actively transport or synthesize all the necessary metabolites, growth factors, ions, everything needed for germ cell survival and differentiation. They even secrete things like transferrin to provide iron.
Function 2, secretion both exocrine and endocrine.
Yeah, exocrine first.
Right.
They constantly secrete fluid, mostly water, into the tubule lumen. This creates the flow that helps flush the sperm out of the testis, and crucially, they secrete androgen-binding protein, ABP.
ADP. Why is that needed if Leydig cells are making testosterone right next door?
Because while testosterone is essential for spermatogenesis, especially the later stages, the levels just diffusing from the interstitium aren't quite high enough within the tubule lumen for optimal spermiogenesis.
So ABP traps it.
Exactly. ABP secretion is stimulated by FSH from the pituitary. It binds testosterone within the tubular fluid, concentrating it to the very high local levels required for that final sperm maturation process. It ensures a supersaturated testosterone environment right where it's needed.
Okay, and their endocrine role, negative feedback.
Yes, they produce inhibin. It's a glycoprotein hormone that travels to the anterior pituitary and specifically tells it to dial back the production and release of FSH.
So Leydig cells respond to LH for testosterone, Sertoli cells respond to FSH for ABP, and make inhibin to control FSH. Nice feedback loop.
A very elegant regulatory loop controlling the overall rate of sperm production.
And there's that fetal role again, too, for Sertoli cells.
Critically important. Fetal Sertoli cells secrete Mullerian-inhibiting substance, MIS, also called anti-Mullerian hormone, AMH.
And this does what?
It causes the regression of the embryonic Mullerian ducts. In the absence of MIS, these ducts would develop into the fallopian tubes, uterus, and upper vagina. So, MIS secretion by fetal Sertoli cells is absolutely required to ensure the development of a male internal reproductive tract.
Testosterone builds the male tract. MIS removes the potential female tract.
Essentially, yes. Both are needed.
And the third general function, phagocytosis.
Simple housekeeping, really. Remember those residual bodies, the blobs of excess cytoplasm shed by maturing spermatids?
Yes.
The Sertoli cells engulf and digest them using their lysosomes. Cleans up the debris, recycles components, and importantly prevents any potentially antigenic germ cell material from leaking back across the barrier.
Okay, sperm are formed, structurally complete but immobile, sitting in the seminiferous tubule lumen. How do they get out of the testes? Through the intratesticular ducts.
Right, it's a specific pathway. First, the ends of the seminiferous tubules narrow into short, straight tubules, which are initially lined just by Sertoli cells. These straight tubules all converge and empty into the rete testis. This isn't really a tube, more like a labyrinth, a network of interconnected channels within that dense connective tissue of the mediastinum testis, lined by simple cuboidal epithelium.
From the rete testis maze, where next?
The fluid and sperm drain from the rete testis into about 15 to 20 efferent ductules. These pierce the tunica albuginea and lead out of the testis proper.
And these efferent ductules have a unique look, don't they?
Very characteristic histology. The lumen has a scalloped or wavy appearance.
A scallop.
Because the lining alternates between patches of short, non-ciliated cuboidal cells and patches of taller, ciliated columnar cells.
What do these different cells do?
The short, non-ciliated cells are absorptive. They reabsorb the vast majority of the fluid that was secreted by the Sertoli cells back in the tubules. This concentrates the sperm significantly.
And ciliated cells.
Their cilia beat in a coordinated way, creating a current. So the combination of fluid absorption pulling things along and the cilia actively sweeping creates the flow that moves the sperm out of the testes and towards the next structure.
Which is the start of the excretory genital ducts. The epididymis?
Yes, a single, incredibly long, highly coiled tube. If you stretched it out, it'd be nearly 6 meters long, but it's packed into this comma shape on the posterior testes.
Divided into parts.
Anatomically, yes. The head, which receives sperm from the epiductules, the body, where most of the action happens, and the tail, which is the primary storage reservoir for mature sperm before ejaculation.
What's the lining like in the epididymis?
It's pseudostratified columnar epithelium. The main cells, the principal cells, have these very long branched microvilli on their surface called stereocilia.
Stereocilia. Are they modal like cilia?
No, they're non-modal. They function primarily to increase the surface area because the principal cells are very active in both absorbing residual fluid and secreting various substances into the lumen that are crucial for sperm maturation. You also have small basal stem cells.
Okay, so sperm spend quite a while here. Like weeks.
Yeah, transit time is roughly 2 to 4 weeks. And this journey is absolutely critical. It's where sperm go from being structurally complete but non-functional to being potentially fertile. It's maturation central.
What key changes happen during this epididymal transit? The sources list three major ones.
Okay, first, they acquire the potential for forward motility. They gain the biochemical machinery to swim progressively, although factors in the epididymal fluid mostly keep them quiescent here to conserve energy.
Makes sense. Second, final modifications to the acrosome occur, sort of fine-tuning its structure and enzyme content.
And third, maybe the most crucial one, membrane changes. Yes, this is vital for controlling when fertilization can happen. The sperm cell membrane undergoes significant reorganization. Cholesterol is added, making it less fluid, and specific proteins and glycoproteins, sometimes called decapacitation factors, are coated onto the surface.
Decapacitation factors, what do they do?
They stabilize the sperm membrane and, importantly, they inhibit the acrosomal reaction. They put the brakes on, preventing the sperm from prematurely releasing its enzymes while it's still in the male tract or even immediately upon entering the female tract.
So the epididymis both enables fertilization potential, motility acrosome, and prevents it from happening too soon, decapacitation.
Exactly. True fertilization competence requires these factors to be removed later in the female tract through a process called capacitation. The epididymis sets them up but keeps them safely inert.
Clever. Okay, from the epididymis tail, during ejaculation, sperm are propelled into the ductus deferens, or vas deferens.
Right, a long, relatively straight muscular tube that travels up through the spermatocord into the pelvis.
And this structure screams propulsion, right? Thick muscle.
Incredibly thick muscular wall relative to its small, often star-shaped lumen. And it's not just thick, it's organized into three distinct layers of smooth muscle: inner longitudinal, middle circular, and outer longitudinal.
3 layers. That's unusual. Why so much muscle?
For generating extremely powerful, rapid peristaltic contractions during ejaculation, this muscular arrangement allows it to forcefully propel the sperm stored in the epididymis tail all the way up and towards the urethra in a fraction of a second.
Okay. The vas deferens travels in the spermatocord, then loops over the ureter, widens into an area called the ampulla near the bladder.
Correct. The ampulla is where it joins with the duct draining the seminal vesicle.
To form the short ejaculatory duct.
Yes, which then passes through the prostate gland to empty into the prostatic urethra.
And because the vas deferens is relatively accessible in the scrotum, that's the target for vasectomy.
Exactly. Surgical sterilization involves cutting or blocking the vas deferens. Sperm transport is interrupted.
What happens to the sperm still being produced? Do they just back up?
Production continues normally in the testes, and maturation starts in the epididymis, but the sperm that reach the blockage site just degenerate. They're then phagocytosed and removed by macrophages within the epididymis and vas deferens. The body cleans them up.
Is vasectomy reversal usually successful?
It can be. Surgically reconnecting the tubes is possible, but restoring fertility isn't guaranteed. The sources note that sometimes the chronic blockage and potential inflammation after vasectomy can cause subtle but permanent changes to the lining and function of the epididymis.
Ah, so even if the plumbing is reconnected, the environment might not be right anymore.
Potentially, yes. The delicate epididymal environment needed for that final sperm maturation might be compromised, reducing the chances of successful fertilization even after reversal. #tag V, accessory glands and semen production.
Okay, we've followed the sperm journey up to the ejaculatory ducts. Now they need to mix with fluids from the accessory glands to form the final product.
Semen. Right. Semen isn't just sperm, it's mostly the secretions from these glands. This fluid provides nutrients, buffers, enzymes, factors that activate motility. Basically everything the sperm need for their journey in the female tract.
Let's start with the biggest contributors by volume, the seminal vesicles. They provide something like 70% of the ejaculate.
That's the figure, yeah. Structurally, they're paired, highly coiled tubular glands. Their internal lining, the mucosa, is thrown into these incredibly complex, thin folds.
By the folds.
Massively increases the surface area for secretion. The lining is simple or pseudostratified columnar epithelium packed with secretory cells, and the whole thing is wrapped in smooth muscle for forceful contraction during ejaculation.
What's in their secretion? It's described as yellowish, viscous.
And its production is totally dependent on testosterone. Key components are, first, fructose.
Sugar.
For energy.
Exactly. It's the primary fuel source for sperm motility. They need a lot of energy for swimming.
Second, prostaglandins.
What do?
They're thought to stimulate muscle contractions in the female reproductive tract, the uterus, the fallopian tubes, potentially helping sperm travel upwards towards the egg.
Okay. And third, fibrinogen, like in blood clotting.
Yes, the same molecule. Seminal vesicle fluid contains fibrinogen. This allows the semen to coagulate to form a temporary clot or plug right after ejaculation within the vagina.
Why clot? It seems counterintuitive.
It's thought to help keep the semen mass together initially, preventing immediate loss from the vagina and maybe providing some protection for the sperm until other enzymes can liquefy it again.
Okay, we'll get to those enzymes. Next gland, the prostate, the central one surrounding the urethra.
Right, a dense, roughly walnut-sized organ made up of 30 to 50 individual tubuloacinar glands. These glands are embedded in a distinctive fibromuscular stroma connective tissue mixed with smooth muscle fibers.
The sources emphasize understanding the prostate zones. This is clinically vital.
Absolutely paramount for understanding prostate pathology. There are 3 main zones.
Let's start small, the transition zone.
This zone makes up only about 5% of the gland volume and directly surrounds the prostatic urethra just below the bladder. It contains small mucosal glands. Why is it important? This is the primary site for benign prostatic hyperplasia, BPH.
The common enlargement with age.
Exactly. Because it surrounds the urethra, when these glands enlarge in BPH, they squeeze the urethra and cause those characteristic urinary problems, difficulty starting, weak stream, etc.
Okay. Then the central zone.
About 25% of the tissue surrounding the ejaculatory ducts as they pass through the prostate contains submucosal glands, less commonly involved in major pathology.
And the biggest zone, the peripheral zone.
Makes up about 70% of the gland, contains the main prostatic glands and forms the outer part of the gland, especially the posterior and lateral aspects. This is clinically vital because it's the predominant site for prostate cancer, adenocarcinoma.
And its location is why a rectal exam can sometimes detect it.
Precisely. The peripheral zone is right up against the rectal wall, so a digital rectal exam can potentially feel nodules or hardening in this zone, prompting further investigation.
What does the prostate secrete into the semen?
A complex, slightly alkaline fluid. Contains various things like citric acid, zinc, enzymes like acid phosphatase, and importantly, prostate-specific antigen, PSA.
PSA. We hear a lot about that. It's a protease, an enzyme.
Yes, a 34 kilodalton serine protease. Its main job in semen is to break down that clot formed by the seminal vesicle fibrinogen.
So seminal vesicles clot it, prostate liquefies it.
Right. PSA acts over maybe 20-30 minutes after ejaculation to liquefy the coagulated semen, which allows the sperm to become motile and begin swimming out into the female tract, a timed release mechanism.
An elevated PSA in the blood is used as a cancer marker.
It can be an indicator, yes.
Yeah.
Normally, very little PSA leaks into the bloodstream, but if the glandular structure is disrupted, like by cancer, BPH, or even inflammation, prostatitis, more PSA can leak into the circulation. It's not cancer-specific, but it's a valuable tool for detection and monitoring.
One more thing in the prostate. Corpora amylacia. What are those?
Little laminated concretions, often calcified, found within the lumens of the prostatic glands. They're made of precipitated glycoproteins and keratin sulfate. They become much more common with age.
Clinically significant.
Nope, just a normal finding, especially in older men. No known significance.
Okay. Last accessory glands. The tiny bulbourethral glands, or Kelper's glands.
Yeah, pea-sized glands located down in the urogenital diaphragm with ducts that empty into the very proximal part of the penile urethra.
What's their contribution?
They produce a clear, viscous, mucus-like secretion. This is often released before ejaculation during sexual arousal.
Like a pre-ejaculate.
Exactly. Its main job is thought to be lubrication of the urethra and neutralizing any residual acidic urine that might be hanging around, creating a more hospitable pathway for the semen that follows.
All right, we've manufactured the sperm, matured them, mixed the semen. Now for the delivery system. The penis.
Yeah.
Structurally, it's based around 3 columns of erectile tissue.
That's right. You have the 2 larger dorsal columns, the corpora cavernosa, CC. These are the main erectile bodies responsible for rigidity.
And the third one.
The single ventral column is the corpus spongiosum. It surrounds the urethra, keeping it open during erection, and it expands at the tip to form the glans penis.
And all 3 are wrapped in.
A very strong, dense fibroelastic sheath called the tunica albuginea. Similar name to the testes covering, but here it plays a crucial role in the mechanics of erection.
What's the tissue inside these corpora actually like? It's not solid, right?
No, not at all. It's basically a sponge. It consists of numerous irregular interconnected vascular spaces like caverns or sinusoids lined with vascular endothelium.
Separated by?
Walls called trabeculae. These trabeculae contain connective tissue, but also, crucially, a lot of smooth muscle fibers. Controlling the tone of this smooth muscle is the key to erection.
How does blood get into these spaces, the vascular supply?
The main supply comes from the central arteries, which run down the middle of each corpus cavernosum. These branch into smaller, highly coiled arteries called helicine arteries.
Helicine, like a helix or coil?
Exactly. When the penis is flaccid, these helicine arteries are constricted, limiting blood flow into the cavernous spaces. They empty directly into those spaces.
Okay, let's walk through erection. It's under parasympathetic control initially.
Primarily, yes. A sexual stimulus, physical or psychological, triggers parasympathetic nerve signals from the spinal cord.
What neurotransmitter is released?
Acetylcholine, ACH. It's released near the smooth muscle of the trabeculae and the helicine arteries. But AC doesn't directly cause the massive relaxation needed. It acts on the endothelial cells lining those blood spaces and arteries.
Ah, the endothelium is the middleman.
Precisely. The endothelial cells, stimulated by AC, release the key signaling molecule, nitric oxide, NO.
NO, the vasodilator.
The critical vasodilator here. NO is a gas. It diffuses rapidly into the nearby smooth muscle cells. Inside the smooth muscle, NO activates an enzyme called guanylate cyclase.
And guanylate cyclase makes.
Cyclic GMP, or cGMP. This is the intracellular messenger molecule that actually causes the smooth muscle to relax.
So, stimulus parasympathetic nerves, OKI endothelium guanylate cyclase, cGMP, smooth muscle relaxation.
That's the cascade. And when the smooth muscle in the walls of the helicine arteries and the trabeculae relaxes, resistance to blood flow plummets.
So blood rushes in.
Exactly. The helicine arteries open wide, blood pours into and engorges the cavernous spaces, filling them up like a sponge soaking up water.
But filling them isn't enough for rigidity. How does it get hard?
Hmm.
That involves the tunica albuginea again, right?
Absolutely critical. As the corpora cavernosa rapidly expand with blood, they press outwards against that tough, relatively inelastic tunica albuginea.
Okay.
This outward pressure physically compresses the veins that normally drain blood out of the corpora, squeezing them shut against the tunica.
Trapping the blood inside.
Precisely. Massive inflow combined with drastically reduced outflow leads to a rapid increase in pressure within the corpora cavernosa, causing tumescence and rigidity. It's a veno-occlusive mechanism.
Brilliant.
Yeah.
How does it reverse? Detumescence and often ejaculation involves the sympathetic nervous system.
Yes. Sympathetic nerve stimulation takes over. It causes the smooth muscle in the helicine arteries and trabeculae to contract strongly.
Cutting off the inflow.
Right. Blood inflow decreases dramatically. As the pressure inside drops, the compression on the draining veins is released, blood flows out, and the penis returns to a flaccid state.
This mechanism is central to understanding erectile dysfunction, ED.
Definitely. ED can have many causes: vascular disease affecting inflow, diabetes causing nerve or blood vessel damage, psychological factors, medications. But many treatments target this final pathway.
Like sildenafil and similar drugs, PDE5 inhibitors.
Exactly. Remember cGMP, the molecule that causes smooth muscle relaxation? There's an enzyme in the smooth muscle called phosphodiesterase type 5, PDE5, whose job is to break down cGMP, ending the relaxation signal.
So sildenafil blocks the breakdown enzyme.
Correct. By inhibiting PDE5, drugs like sildenafil allow the cGMP produced in response to NO to stick around longer and accumulate to higher levels. This enhances and prolongs the smooth muscle relaxation, making it easier to achieve and maintain the blood inflow and venous compression needed for an erection, assuming the initial NO signal is still being generated by sexual stimulation.
So it boosts the natural mechanism, doesn't create it from scratch.
Precisely. It works with the natural pathway triggered by arousal. A really neat example of targeted pharmacology. #TagHoutro. So we've really covered the entire system, haven't we? From the intricate molecular dance of meiosis and spermatogenesis, governed by precise temperature control, through the amazing security of the blood-testis barrier, the endocrine control loops, the vital maturation steps in the epididymis, the contributions of the accessory glands.
Right up to the hydraulics of erection. It's all incredibly interconnected.
It truly is. Every part relies on the others functioning correctly.
You know, the detail that really stands out for me as maybe a signifier of true mastery is that paradox of the Sertoli cell barrier. We call it the Titus barrier. Immune privilege is paramount. Yet the Sertoli cells have to actively, temporarily take it apart and rebuild it, just to let those primary spermatocytes sneak across into the luminal compartment.
Ah, the migration step.
Yeah. That managed breach, that moment of controlled vulnerability, that's essential for the whole process to continue.
Yeah.
It just encapsulates the complexity.
It's a beautiful piece of cell biology, coordinating disassembly and reassembly without ever really dropping the shield. You're right, it is remarkable.
Hmm.
And thinking about that level of precision, that essential vulnerability perhaps, brings us back to where we started and to a final thought for you, the listener, to mull over. We spent a good amount of time on that absolute necessity for the testes to be cooler, that 34 degrees C rule. Sperm production hinges on it? So here's the question. Given the intense evolutionary pressure for reproductive success, why hasn't this crucial process adapted to work efficiently at the stable, protected core body temperature of 37 degrees C? What fundamental biological constraints, anatomical, perhaps genetic, maybe biochemical, have prevented mammals from bringing this vital factory fully indoors over millions of years? Why the continued reliance on this external, somewhat vulnerable temperature regulation system?
Hmm. That's a deep question. Why couldn't evolution solve that problem?
Something to think about, reflecting on the intricate systems we've explored today. What limitations might be at play?